Hey y'all, Miss Mayhew here, and we are going to go over the FRQ practice for cystic fibrosis. So the prompt says that cystic fibrosis is a genetic condition that is associated with deficits in the CFTR protein. The CFTR protein is a gated ion channel that requires ATP binding in order to allow the chloride ions to diffuse across the membrane. Now, they also include a diagram for reference of a cell and some of the organelles labeled inside of it. Part A asks you to identify the type of organism that cystic fibrosis would affect. Now, looking at the diagram, I can see that there is a nucleus and there are other membrane-bound organelles like the mitochondria and the Golgi apparatus. So these are things that are only present in eukaryotic cells. So the answer to part A is a eukaryotic organism. Part B asks you to identify the most likely cellular location of the ribosomes that synthesize the CFTR protein. The answer to that is going to be the rough endoplasmic reticulum. That's because although ribosomes that are found free-floating in the cytoplasm and ribosomes found on the rough ER both synthesize proteins, they both synthesize proteins that end up doing different things and going to different places. The ribosomes that are free-floating in the cytoplasm, they produce proteins to be released directly into the cytoplasm to be used like within the cell. While the ribosomes that are attached to the rough endoplasmic reticulum, they make proteins that are going to travel through vesicles where they'll be embedded in the membrane of another organelle or to the membrane of the entire cell to be embedded there or even excreted. Part C asks for you to draw arrows to describe the pathway for production of a normal CFTR protein from gene expression to final cellular location. Now, in order to do this, the first thing we're looking at is gene expression. So we have to start at the nucleus where we have the DNA that is transcribed into RNA, and then it's going to travel to the ribosomes on the endoplasmic reticulum. This is where it's going to be translated into the protein. Then it is going to travel to the Golgi apparatus to be processed and then to the plasma membrane. One important thing to note right here is that in order to get this point, you have to draw the arrow to the plasma membrane. Remember, in the prompt, it tells you that it's a ion channel. Okay, so it has to be embedded into that plasma membrane. If you go past the plasma membrane, that means it's excreted. And that channel doesn't do a lot of good if it's outside of the cell.